Good afternoon, everyone. If you'd like to take a seat, we will make a start on this afternoon's lectures. So for those of you who don't know me, my name is Professor Selina Wray, and I'm one of the Deputy Directors here at UCL Queen Square Institute of Neurology. And it's really my pleasure to give the opening remarks for this evening's inaugural lectures. It's wonderful to see the room so full with so many colleagues, and for those of you who are visiting us here, I really extend the warmest of welcomes. Tonight is a special occasion. Inaugural lectures are where we come together to celebrate an individual's academic and professional achievements. But we also get to find out a little bit more about the person behind the research and the journey that has brought them to where they are now. And also as a community, it gives us an opportunity to reflect on how their work has shaped the Institute and also shaped our field. We're really fortunate today to hear from two colleagues who I've had the pleasure to work with for a number of years now, and both are respected leaders in the field of dementia research. They've made transformative contributions to our understanding of Alzheimer's disease and other forms of dementia. And your presence today is a testament to the outstanding work that they've done at the Institute and the broad impact of these two remarkable individuals. Although it's coincidence, this event is also happening in the same week as International Women's Day, and I think it's wonderful that we have this evening to showcase and celebrate female success, female leadership, and female role models. The theme of International Women's Day this year is Accelerate Action, and I really believe the work of both of our speakers aligns with that theme as they aim to tackle one of the most urgent global health challenges. So turning now to our first speaker, I'm delighted to introduce Jo Barnes, who is Professor of Imaging Neuroscience at the Dementia Research Centre in the Department of Neurodegenerative Disease. Professor Barnes has been a pioneering figure in neuroimaging, leading research that has brought key advances in our understanding of Alzheimer's disease. Her innovative work has significantly advanced how we study brain changes in dementia, and her reputation in this area is evidenced by her funding, over £1 million, her publications, of which there are over 100, and her leadership roles in international consortia and working groups on the study of white matter hyperintensities. But beyond Jo's groundbreaking research, it's really hard to think of many people who have done more at the Institute to support early career researchers and make the Institute of Neurology a supportive environment where junior colleagues can thrive. Professor Barnes has supported colleagues and students in countless ways, both informally and formally. She's been an incredible mentor to many early career researchers, providing guidance, psychological safety, and fostering an environment of collaboration and intellectual curiosity. Formally, Professor Barnes has led the Institute's mentoring scheme for several years. She's currently the faculty graduate tutor, where she has a significant impact on student well-being, supporting 1,000 PhD students across the faculty. An impact that is sometimes difficult to quantify, but expansive and profound. Her dedication to her research and student support is truly exceptional. So please join me in welcoming Professor Barnes to deliver her inaugural lecture.
Thank you ever so much for coming today. It's really nice to see so many people. I didn't think I'd say that, but it is really nice to see so many people. So I'm going to speak first, and then I'm followed by my lovely colleague, Cath. So my talk will be about memory structures and measuring abnormal signals using MRI. So first and foremost, I want to say thank you to my lovely family, my nearest and dearest, my husband Duncan and my kids Ashwin and Aloan, who are in the audience. But luckily for everyone, Dave in the middle is not. He's quite naughty, and Jack would be very noisy. So I'm glad they're not here today, but they're definitely my support companions. I also want to say thank you to my mum and dad. My dad is watching online, and my mum's here in the audience, and also my in-laws. Professor Barnes Senior, who's in the right-hand picture there, and also my mother-in-law, who sadly passed away before Christmas, but I know that she was very proud that there was yet another professor in the family. So work life before UCL. So before I was at UCL, I did an undergrad degree in natural sciences. And I thought actually what I wanted to do was work in science television. So I had a detour between my undergrad and my time at UCL. So this culminated after a few years of working at Tomorrow's World. If you look up Tomorrow's World, this is what you get on the iPlayer nowadays, because Tomorrow's World is for those people who are much younger than me or grew up in a different country, Tomorrow's World was a very long-running magazine science programme that aired on BBC One. And this is actually a programme from the 1960s. It did not look like that when I worked there. We were positioned between Horizon and Crime Watch at White City, and actually it was a really interesting time. So I'm glad I did it, but I definitely knew that whilst I was there, I was more interested in the science than actually doing the filming and the researching. I actually wanted to do the science myself. So I decided to look around for graduate jobs, and I saw one that came up at Queen Square in the Dementia Research Centre. So I applied for the job, and I was interviewed by these three people. Now, I know Martin is not here today, and he doesn't appear in black and white in any way, but it's the best photo. It's the best photo I could find of him. So Martin Rosser on the left-hand side, Rachel Scarhill in the middle, and Nick Fox on the right. Luckily, they gave me and two other people at the same time a job. And actually, these three individuals I was very lucky to have them as my PhD supervisors when I was allowed to do a PhD part-time. So the job that I was actually taken on to do was to look at MRI and make quantifications using MRI. So I know there are people in the audience that don't use MRI or aren't used to looking at it. This is an MRI that we would tend to use in those early days. It's actually my MRI of my brain, so I know it's normal. I think it's normal anyway. This is a bread and butter sequence for image analysis. So just to orient you, on the left-hand side there's the coronal view. This is as if you're looking straight onto an individual and you can see right through their face. And then in the middle is the sagittal view, and it's as if you're looking through their ear. And the axial view on the right-hand side is as if you're looking down through somebody's head at the top. And the reason that this is a really good sequence is it's really easy to actually acquire. It doesn't take too long. You get really good grey-white differentiation. So the grey matter, you can see, is dark grey. It's on the outside of the brain. And the white matter is a sort of lighter grey towards the middle. And the CSF, the cerebrospinal fluid that bathes the brain, is black. And that's in the middle of the ventricles and also around the outside of the brain. This is somebody that's a kind of different image to the previous one. It's somebody who has Alzheimer's disease. And you can see some marked differences there. There's a lot more CSF. So the ventricles in the middle are a lot bigger. The brain is shrunken somewhat. And you can see these structures, which are the hippocampi in the medial temporal lobe, are particularly small. And this is what we expect to see when somebody has a sort of typical presentation of Alzheimer's disease. So that what I mean by a typical presentation is they have significant memory problems as well as other problems associated with that. So the kind of job that I and others did as research assistants was to measure the brain. So this blue region around the outside of the brain is called a segmentation, and we would do this for two purposes. One, to measure the volume, because that's what these images are really good for, to measure volumes, but it's also really important in the image processing of the images as well. Not only did we measure volumes of the whole brain, but that medial temporal lobe is really useful to measure, and that's because these structures are disproportionately affected in things like typical Alzheimer's disease. So here we can see a blow-up image of the medial temporal lobe, and the hippocampus has got the H in it. Above that is the amygdala, and underneath the EC is the entorhinal cortex. So one particular study that I published as part of my PhD was this one, where I looked at the volumes of the hippocampus and amygdala in a group of controls, Alzheimer's disease and frontal temporal lobar degeneration. So the FDLD group is a little bit different from the AD group. It's actually a group of neurodegenerative conditions, and they have characteristic presentations. And in terms of MRI, they tend to have atrophy towards the front of the brain. And you can see here On the graphs on the left-hand side, you can see that the hippocampus here in the diamonds has a characteristic atrophy pattern from controls to AD to FDLD, where the AD group is in between the two. And that's true on the left-hand side, which is at the top, and on the right-hand side at the bottom. And why this was really important for us to look at was that at this time, in 2006, in the middle of the 2000s, there wasn't really any good tests for Alzheimer's disease. We have some tests nowadays, but at that time we didn't. And early in the disease course, it was actually quite difficult to differentiate between Alzheimer's disease and FTLD. On the right-hand side here, we actually looked at a bit more detail about the atrophy pattern from the back of the joint, amygdala and hippocampal region, going from the back of the hippocampus all the way through to the front of the sort of amygdala and hippocampus. And what we can see here is that the areas per slice of the brain can show that the hippocampus is really affected in these two groups here. And these two groups are part of that FTLD group. So that this is formed of the frontal variant FTD and then the semantic dementia cases. And the semantic dementia cases are particularly interesting because on the left-hand side, which is the top here, you can see that they are particularly small. So as I mentioned before, we didn't just draw around brain regions for fun, although it can be fun. We actually use those regions to put into an image processing pipeline. And one of the things that we really need to do is to look at change over time. Change over time is a very helpful thing to try to understand whether something is progressing in an individual, the disease is progressing. But when you have somebody come back over a period of time, you can't necessarily put them in the scanner in exactly the same position. You have images like this that are quite hard to compare. But if you align those images using a registration algorithm, then what you can find is that you can generate difference images, which is shown at the bottom here. So because in every image, it's basically just data, so you can do maths on those images and you can show the difference between those two. And because we actually co-registered the brains rather than the whole image, you can see that there's a bit of a mismatch here between the scalps, but we're not really worried about that. You can see the scalp here, and that's quite obvious. But around here, around the ventricles, you can see white, and that's because there's been a change in the volume of the brain, and this is the shift between the two brain volumes over time. And by quantifying that, we can actually look at and actually measure the brain volume loss. And this is represented here by the red line around the ventricles and around the temporal lobe here. So just having a little look at registrations themselves. So this is an individual that was recruited into a North American study. And if I flick between the images, you can see that even without quantification, the registration process is actually really helpful. It can tell you quite a lot of what's going on. Now, this is a really quite extreme example, and the interval between the images is about 60 months, so there's a lot happening. And actually, this individual had multiple scans that are all co-registered here on this slide, and you can see at the top, the first 4 images are actually over the first 12 months, and they were considered a normal control over this time period. It's only when the individual came to 24 months that they were considered to have what's called mild cognitive impairment, where you can actually measure that there are memory differences in this individual compared with a normal group. By 60 months, this individual actually met criteria for AD. But what this shows is the power of registration, even just visually, to actually understand that there is progression in individuals, and you can see that before somebody becomes demented. So this is another case that we actually saw here at the Specialist Cognitive Disorders Clinics in Queen Square. So this is a case that was written by Hilary Archer, and I helped with the imaging when I was leading up to my PhD. So this individual could recognise that they couldn't any longer think 7 moves ahead in chess when they came to the clinic. They could only think 3 moves ahead in chess, and that was that was detectable for them. But obviously, when it comes to assessing whether they deviated from normal, they were absolutely considered within the normal range, not just at baseline, but also at 9 months. But by 2 years, in terms of their cognition, they were considered mildly cognitively impaired. And you can see, actually, at this time point, there is actually a bit more space around the hippocampus here, and this sulcus is starting to open up. So leading up to even mild cognitive impairment, you can see changes in the hippocampus and in the brain using imaging. And by 3 years, this particular individual was only on the cusp of meeting criteria for dementia. Unfortunately, they died of something completely unrelated, and they kindly donated their brain to Queen Square Brain Bank. And it was discovered that they had quite extensive AD pathology. So again, we know that in cases of AD, we know that we can see during life a lot of change leading up to dementia and the diagnosis of dementia, even in people who are really high functioning in the first instance. So Basil Reeder and I also took serial images and measured lots and lots of hippocampi manually. It's quite a hard job. It takes a lot of time. But we did this in a familial AD study. So all the red lines here are people who have familial AD mutations, so mutation carriers, and the blue lines are controls. And you can see that the people with mutations have their volumes of their hippocampi go down over time. And the x-axis along the bottom here, for those that are particularly interested, is time to diagnosis for AD. So when it's 0, that's when the diagnosis of AD can be made. So you can see that actually, even well before somebody gets dementia, you can measure changes in that structure. And more than that, working with the statisticians at the London School of Hygiene and Tropical Medicine, particularly Chris Frost, we measured whether or not, exactly mathematically modelling when we could see those changes and when they deviated from 0. And we know that hippocampi in this FAD cohort can be measured to be different from 0 between 5 and 6 years before somebody gets demented. So one of the other studies I was particularly interested in looking at was a study where there was shorter interval imaging. And the reason why shorter interval imaging might be useful is that in clinical trials and studies, you might want to see whether there's an early signal, whether whatever intervention you have is actually making a difference. So I drew around lots of hippocampi again and looked at 12-month interval imaging and 6-month interval imaging just to sort of see whether or not you could see those changes early on. And indeed you can, although the spread in 6-month interval imaging is slightly bigger. It might be that actually you can detect early changes very quickly in some individuals and in studies that way. And it's really useful to know that this is possible. And again, working with statisticians at London School of Hygiene and Tropical Medicine, we looked at what the average rate of atrophy in the hippocampus is likely to be in Alzheimer's disease participants. So this is a meta-analysis. So that means trawling through loads of literature, getting in contact with loads of people that have published studies to find out whether or not you've missed any work and whether there's any overlap in participants between studies. And actually, at this time, there weren't that many studies looking at rates of change in the hippocampus in Alzheimer's disease, which is kind of odd to consider now because there's so many papers. There's ones that come through in my inbox every day, pretty much, with a new study looking at rates of atrophy. But we quantified that actually the rate of atrophy is between sort of 4 and 5% for Alzheimer's disease participants. And the difference between Alzheimer's disease and control subjects is just over 3%. So it was a useful study. And the reason I've included this one is because when I first tried to publish this particular paper, it was sent off to a journal, and I got a really quite interesting but kind of cutting remark from one of the reviewers that said it was a well-crafted arrow that missed the target by some margin. But, which I think, if you're going to reject a paper, then do it with style. But But actually, I've included it because it did get published, obviously, and it's been cited hundreds of times, so there we go. Well crafted and not missing the target. So I was particularly interested in whether or not you could make advances to measuring the hippocampal change. And one of the reasons why I thought this could be possible was because when you look at brain registrations of people who have Alzheimer's disease and you look in the area of the medial temporal lobe, it's really obvious that there is a shift in the boundary of that hippocampus. And you can see that there is CSF space right next to it. So that's the temporal horn. And so I performed a little study, which you can see here on the left-hand side, seeing whether or not I could apply the BSI to the hippocampal region. And this is just using the baseline hippocampi. And in fact, it did do a pretty good job at quantifying change. You still need that baseline hippocampal region that you draw around, but you're cutting in half, if you've got 2 time points, the number of hippocampi you have to actually draw around, which was it takes at least 45 minutes per hippocampus, so it's quite a lot of manual work. So it did do a really good job, and actually the manual results look pretty similar in controls and AD to the results that you get using the BSI. And Rachel Scarhill and I also looked at 2 different types of non-linear registration to measure the hippocampal changes, and these are shown here in terms of fluid propagation and Jacobian changes, and both of those also showed very similar gains that could be made, and that you just measure the baseline region and then you can actually measure change using that baseline region and registration processes. So all of these techniques showed promise. But I then thought, well, I don't really want to measure the baseline hippocampi either, to be quite honest. I'd rather do it automatically. So with this sort of what looks like quite a complicated set of registrations, I managed to get the average hippocampus from a particular study. And when I say average, I mean I literally looked at the volumes, created the average, and then tried to find the person that was closest to that average, and then used that and transformed it onto the baseline using registration of every individual in that study. And then I measured the change in this particular in this particular study in controls and AD again, and the automated BSI, which is in the black dots here, give very similar results to that of the hippocampal BSI that uses the baseline region and the manual results. So I was pretty pleased with myself actually at this point. But then I got to thinking, well, you know, we've got loads of hippocampi that we've segmented, takes a lot of time. There's no way that the average one is going to be the best estimate of the hippocampal region. You could actually use it a bit like facial recognition and flip through loads, a bit like a Rolodex, to find the best possible matched hippocampi from a library of subjects. So that's what I did here. And with a number of different, again, registration procedures, I managed to actually estimate that baseline region in a much better way and generate rates of change, which again, we looked at using 3 different methods, BSI and then the 2 nonlinear techniques. And they all gave good results. But then along came Kelvin, who was a fantastic postdoc that I had pleasure of working with. And we thought, well, why don't we fuse a number of hippocampi together? So that was the next step. So we actually ran a study and worked out that if we took the top 8 subjects that matched any particular subject that we wanted and fused the regions together, we could not only create really good regions and estimate volumes really well, but we could also generate rates of change. So this was a big study, a bigger study than we'd done before. So we had that in controls and in Alzheimer's disease, and we had the characteristic pattern that we would expect to find in these groups, where the mild cognitive impairment group, which is the group where they've just got potentially memory problems that are measurable but they don't fulfill dementia criteria, they sit in the middle, and they do that for both the volumes and also the rates of change. And then moving on to excellent students that I've had the pleasure of working with, Emily took our techniques and asked different questions with the hippocampi. So in particular, her question was whether or not having an ApoE4 allele, so that's a particular, it's one of the strongest risk factors for sporadic AD, a genetic risk factor. It's not familial AD, but it's the strongest, one of the strongest risk factors for sporadic AD, and whether or not that changed the rate of atrophy in the hippocampus in a big study. And what she showed is in the mild cognitive impairment groups and also in AD, you get a higher rate of atrophy in those individuals who have that ApoE4 allele, not in the controls. And she also looked separately at those who remained stable, MCI, MCIS, and also those who progressed to AD. Another thing Emily did in her PhD, which was excellent, was looking at atypical AD. So there's a number of different variants of atypical AD. So if we consider typical AD being more the memory-led AD, posterior cortical atrophy is a very different presentation. So these are images, the MRIs of somebody with PCA, and you can see that actually it's an axial view. The back of the brain at the bottom there has an awful lot more atrophy. And these individuals often present not with memory problems, but they might go to their optician and describe having problems with their vision. But we wanted to know in this group, is there any differences at all in the hippocampi? You wouldn't necessarily expect it because it's not a memory-led variant of Alzheimer's disease. But you can see in the top left-hand side picture where the red arrows are pointing towards, that there was actually a bit of a difference at the back of the hippocampi of those people with PCA on average. So it's kind of as you might expect if you were going to say there's an area affected here, it's towards the back of the brain or towards the back of the hippocampus, but it's nowhere near as extensive as the typical ADs, which can be seen at the bottom there. So the blue at the bottom here shows extensive changes in the hippocampus or differences compared with controls. So a number of the students that I've had pleasure of working with have not just looked at region of interest studies, but they've looked over the whole brain. There's a number of different ways you can do this. I'm showing here some surface-based techniques on the left-hand side, which is cortical thickness analyses. So this asks the question whether or not the cortex is thinner or thicker all the way through the brain, related to either being part of a group or here, how it changes or it's affected by age. So sadly for everyone in the room, the older you get, the thinner the cortex across the whole cortex. And similarly on the right-hand side is a voxel-based type of analysis that's similar. And this shows that as age goes up, the volume throughout the brain of the grey matter goes down. And Manya Lehman, who was another excellent student that I worked with, used cortical thickness analyses to look at both typical AD and atypical AD, so a group of atypical presentations of Alzheimer's disease. So, and she compared these to controls, and her major question here was, is there an area where both typical and atypical AD are different than the controls? So we see in the top right, under A, there's extensive differences across the whole cortex in terms of thinner cortex in those people with typical AD. But it's far more restricted under B in the atypical AD subjects. So the frontal pole right at the front here, the posterior cingulate precuneus here, and the medial temporal lobe being areas that are affected. And when you look at the overlap of those regions, these orange bits are where both typical and atypical AD are different than controls. Manya also went on to look at the PCA, one of the subgroups of atypical AD, and she looked at whether or not there was change over time in the grey matter in PCA compared with controls. And what you can see here, you kind of expect with the PCA, you'd have a posterior gradient because this is what the scans look like when people are imaged at first time point with PCA. a lot of atrophy at the back, and you expect that to continue. But in fact, what she showed is that actually you get grey matter change throughout the brain, which looks very similar to the typical AD changes that you can see there. So just because somebody presents with a very sort of atrophic pattern in a particular area doesn't mean that the atrophy stays in that particular place, which is important to know. And Cassie Fyford, who I supervised more recently and had the pleasure of working with her for a number of years, she used the grey matter changes across the whole brain to ask quite a different question, which was, where is the relationship with age different according to group? The groups that she was interested in, controls, MCI and AD. And what you can see here is there are some areas that differ in terms of that relationship. And by extracting the volume change in each voxel in these areas, you can see what that pattern means. And so in controls, in both the left angular gyrus and left precuneus, as you get older, you get more grey matter change, so the slope goes down. But the opposite is true in MCI and in particular in AD. And what we think that means is that those who present younger, who have MCI or AD, are much more likely to progress quickly. It's a more aggressive disease. And Nicola Hobbs, who I worked with quite some time ago, looked at grey matter change, white matter change and CSF change in early HD. So HD is a Huntington's disease, and Huntington's disease is a different neurodegenerative condition. And she showed that there's extensive grey matter change, but in particular white matter change in this group of individuals. And the green arrow there shows the core date in the top left-hand corner. And, you know, as much as you don't want anything to light up in some ways when you're doing analyses, it's where you might expect there to be a lot of change in Huntington's disease, which is somewhat reassuring. Nikki then went on to actually look at region of interest analyses in the core date. And much like the FAD study that I showed you earlier that Basil and I did, this particular study shows that the controls at the top, there is a bit of a downward trajectory in terms of their core date volumes with age. But in terms of the Huntington's disease and pre-manifest converters, who are people that have the gene for Huntington's and convert during the course of the study, they have changes in the core date which are quite profound, and you can see that they actually start on average from a much lower place in terms of their volume than the controls. And this is also evident when you look at disease duration and plot the core date volume with disease duration. And finally, for Nikki's work, she looked at core date BSI because, you know, it actually works in lots of areas. And Nikki showed really nicely in her work that you could calculate the core date atrophy rate using the BSI, which was a really excellent development. So we thought we'd made quite a lot of gains with the hippocampus in particular, and measurement of the hippocampus. So I was sort of looking around for something else to do, and what caught my eye was cerebrovascular disease, which is slightly different. And working with a lovely colleague of mine, Carol Sudra, who's here today, I'm very lucky to have Carol here today, we started investigating white matter hyperintensity, which you can see here as the second the first red box there, and also cerebromicrobleeds, which is towards the right. So this really took my eye because actually within the context of Alzheimer's disease, most people when they come to postmortem will have some cerebrovascular disease. The co-pathology percentage is really high, so I wanted to understand it in a much more detail, and the imaging has come on hugely in that regard, so it's actually easier to study it as well. So just to show you what white matter hyperintensities look like, this is an axial flare image on the left-hand side, and you can see the red line around the white matter hyperintensities. They're really bright. That's why they're called white matter hyperintensities. And this is a flare image, and the co-registered T1 is next to it. And the T1 image, which is the images that I've shown you mostly up to now, you can see that that region's actually dark. So it's not like they're invisible on T1 imaging, but they're really, really obvious on this flare imaging, and so they're easy to measure. Microbleeds, however, are somewhat different. You need a different kind of imaging again called T2 star. There are a number of different types of imaging that can visualise these microbleeds. And they're the dark spots that you can see on the left-hand side image. They're not lesions in the typical sense that, you know, they're as big as they look there. That's actually blooming artefact. It's a signal void. So it's a little bleed that's come from a vessel, and the iron within the blood that's deposited actually gives this sort of signal artefact or void. And it's usually very spherical like that. And rather than create a volume from these, what we tend to do is we tend to count them, which is quite a lot of work. So sometimes some individuals have hundreds, and it can take many hours, but many people don't have any at all. So then a student, Phoebe, came to work with Tammaryn and me, and she also worked with Carol as well, and she wanted to investigate the white matter hyperintensities in more detail. She'd done some work on that already. And within the context of AD, the first question you might well ask is about the amyloid protein, so amyloid beta. And the reason is because that's what we think is one of the earliest proteins to be deposited in the brain with respect to Alzheimer's disease. It's a diagnostic hallmark protein. So rather than actually measuring that directly in the brain, Phoebe looked at CSF A beta 1 to 42. Now, as amyloid beta is deposited in the brain, the actual CSF values go down, so there's an inverse relationship there. And so when Phoebe looked at the white matter hyperintensities and how they related to this CSF A beta 1 to 42, she found a consistent relationship across groups. I know there's quite a lot of scatter here, but it's statistically quite a consistent relationship. So she found that as there was more deposition, i.e. lower CSF A beta 1 to 42, there was more white matter hyperintensity volume. And that was seen in controls in a group here called subjective memory complaints, which is the SMCs. And that's a bit like the chess player that I mentioned earlier. It's people that have these memory issues, but they're not really measurable. They perceive that they've got them, but they're not measurable on tests. And then there's two groups of MCI, early MCI and late MCI, and then Alzheimer's disease. So we thought, well, that's interesting. We should look a bit further. So we looked at a blood-based biomarker for neurofilament light. Now, neurofilament light is a neurodegeneration biomarker. So more of that in your blood would indicate more neurodegeneration and potentially axonal degeneration. And the axons being in the white matter would make sense. If you've got more neurofilament light, you'd get more white matter hyperintensities. And that is what Phoebe showed, but in particular in the AD, the MCI groups and the SMCs, but not in the controls. And more importantly, in this particular study, she found that that relationship was age dependent. So as soon as you adjust for age, there's no relationship at all. And we think that that's because age explains both white matter hyperintensities and the neurofilament light levels. And then Cassie went on and looked even further at how white matter hyperintensities relate to brain atrophy rates at the top and also hippocampal atrophy rates. So she showed that in the whole brain, in controls, white matter hyperintensities were related to the whole brain rates, and in the controls and MCI, the white matter hyperintensities were related to hippocampal rates. And if you adjust the association of the hippocampus for whole brain rates, you still see that relationship in the hippocampus. So we know that it's disproportionate in this particular area. So the hippocampus is vulnerable to something that is also related to these white matter lesions. Now, it's not a relationship we totally understand yet, but we're still working on this at the moment to try to understand what's going on. And Tom Brown, who's a current student who is working with Carol and me, is looking at this relationship in a bit more detail. So he's looking at a birth cohort. So these are people born in 1946, and they're all cognitively normal on this graph. And he's looking at how white matter hyperintensity changes. So change over time relates to hippocampal atrophy rate. And he has shown that actually it does, the two things do track together. So there is something either causing both or partially overlapping pathological processes here. So Phoebe wanted to look in a little bit more detail about the pathological underpinnings of the white matter hyperintensity. And in order to do that properly, what you have to do really is look under the microscope. So one of the first things that we did, and Phoebe did, was to see whether you could scan half brain in the scanner at post-mortem. So when a brain is donated to Queen Square Brain Bank, what tends to happen is the brain is cut in half and half is fixed. The other half is frozen. And the fixed half we can then take to the scanner, put it in the scanner, and get really nice imaging. So you can see here on the left-hand side, the ex vivo T2 here is the really nice post-mortem imaging you can get. And we did this in 5 subjects, and these were 5 individuals who had Alzheimer's disease and had imaging during life. And through a series of registrations, we were able to actually align the brain images in life to those at post-mortem, which is really useful. And that was actually one of Phoebe's questions: can you do this? Because there's a lot that happens over time between the last scan during life and then actually scanning a half brain after death. But the nice thing is about the registration scheme is once we worked it out, you can actually transform either the imaging during life into the space of the ex vivo T2 or the other way, so that we can look at it in the space of the imaging during life, and particularly the flare imaging. And further to that, Phoebe could look at how sections of the brain corresponded to the post-mortem imaging, and this is work that we did with Eugenia Iglesias, who is based now in the US. And what she and Eugenia were able to do was to examine how this post-mortem image looks compared with some of the stained slices. So this one in particular is really useful to look at. So this is LFB. This stains for myelin, which is what you expect to find in the white matter. That's why it looks white. And what you can see here is an area here around the ventricle of demyelination. So there's not this royal blue colour throughout. And the post-mortem image here, you can see that there is a white matter hyperintensity here. So what Phoebe was able to show with this work was that not only did you get demyelination within these white matter hyperintensities, but you also got a decrease in the vessel density, and the cells that actually do the myelination were fewer in number. Another thing we want to do, of course, is look at what predicts these particular markers of brain health, if we consider them generally as brain health markers. So to do that, you have to look back in the past, and we also want to look into the future. At the end of the day, nobody really cares if they got white blobs on their scan, if it means nothing. So actually trying to understand a bit more about what predicts them and actually what they predict is really helpful. So Chris Lane, who worked with me and a number of people on that birth cohort that I mentioned before that Tom's working on now, looked at both white matter hyperintensities and hippocampal volume. And he looked at all of the 1946 cohort that came back for imaging at the age of 70. And the lovely thing about this cohort is that they've been followed since birth, and they have blood pressure measures throughout life. So they had them at 36, 43, 53, and then about 60, and then 69. And what he showed was that higher blood pressure, both systolic blood pressure and diastolic blood pressure at 53 years, and a rise up to that point, was associated with more of the white matter hyperintensities that you saw later on in life. Further to that, you could see that higher systolic blood pressure age 43 and also rising systolic blood pressure up to that age was associated with lower hippocampal volumes later in life. So although this sort of is, it's very interesting, what this also tells us is that there's a window of opportunity to look after the health of individuals earlier in life to prevent potential brain health problems later on. Chris also looked in a bit more detail at cardiovascular risk and cardiovascular health. And these summary scores were available at 36, 53 and 69. And he showed here that poorer cardiovascular health at 36, 53 and 69 were associated with more white matter hyperintensities later on. and actually the effect was really great at 36, which is a bit disappointing for me, but that time has passed. But and in terms of hippocampal volume, something very similar was seen at age 36 with smaller hippocampi. So again, it tells us that actually looking after ourselves earlier in life and having the capacity to do that is really important, and it might prevent later life poorer brain health. And Lloyd, who was a student that finished quite recently with me, was looking at how these things have an impact moving forward. So what do these things predict? And he looked at a number of different things, a number of different variables to try to understand this, including the white matter hyperintensities and microbleeds, and also whole brain volume and hippocampal volume. And his work showed in a group of controls, it's not the birth cohort, it's a different study, that lower hippocampal volume and more white matter hyperintensities predicted conversion from controls to mild cognitive impairment. And on the right-hand side, you can see the survival graphs for those variables. Now, survival, for those people that don't do this kind of work, it doesn't mean literal survival. This means, in this particular case, conversion to MCI. So 100% is 100% of controls. But also, we know we've got a lot of data nowadays, and actually there are algorithms and that can actually tell us much more about the data than we can just use with normal statistical methods. And actually we can put the data into machines, not this kind of machine, but other machines, and it can tell you patterns about the data itself. And we were particularly interested in this sort of machine learning AI type approach for all the data that we generate. And one of the questions that we were particularly interested in relates to this graph. Now, this is, for those who do Alzheimer's disease, this is a mandatory graph in every presentation, it seems. But the point I'm mentioning of why I'm bringing it into this particular presentation is because it shows how biomarkers go from normal to abnormal and when it happens throughout the disease course. Now, this is just a model, it's a schematic, it's not necessarily true in every case, but it shows amyloid beta, it shows tau, brain structure, memory and clinical function, but there's no cerebrovascular disease in there whatsoever. So we wanted to ask the question about, actually, do you get heterogeneity, do you get differences in different groups in terms of how these things become abnormal in Alzheimer's disease, and also where does cerebrovascular disease sit? So we use white matter hyperintensities again, but also lots of other measures that are mentioned in that particular graph in terms of cognition and so forth. And the white matter hyperintensities are here, and they track along through these groups. So we used a massive data set with lots of controls, MCI, AD, and we had a look at how the algorithm would predict these groups to be and what the likelihood of the ordering of these biomarkers might be. And what you can see is that the algorithm decided that actually there were 4 groups, and white matter hyperintensity was the last event in the biggest group, group 1. But in groups 2 and 3, it was either the third or second thing to potentially become abnormal. In the smallest group towards the right, it was somewhere between 5 and 10 in terms of becoming abnormal in terms of the event position, but that was a really relatively small group. So this tells us that actually, you know, in some individuals it's actually quite a late event, in other individuals it's a really early event, but there's certainly heterogeneity there in terms of the data. Lloyd took this further and sort of asked the question, actually, if we assume that amyloid deposition is the first event, why don't we just take all the amyloid positive people that we can find in this study and see whether white matter hyperintensities comes out as an early position at all, or whether it disappears. And the white matter hyperintensities is in this position in the 3 diagrams here. And the second group down, which is actually the smallest group, has white matter hyperintensities as the first event. So obviously all of these individuals are amyloid positive, and we don't know where the amyloid positivity is in relation to all the other biomarkers here. But we know that actually that there is heterogeneity even if you just take those people who are considered to be on the amyloid pathway. And finally, in terms of research, I wanted to bring it back to the clinic. So Hugh, who was a student that I worked with, who finished relatively recently, did a lot of work on bringing a lot of the volumetric stuff, that we've been talking about this evening, into the realm of the clinic. At the end of the day, I love playing around with data. I love messing around with it. It's great fun. But if it's not going to be useful in the future for patients, then what's the point in doing it? So he looked at how you could actually generate reports that might be supportive of diagnoses in a number of different conditions, including in dementia. And the idea of these kinds of reports is that they don't replace radiologists, but they might support a radiologist's decision or reporting on the imaging. So included in this kind of report may be hippocampal volumes, it's also brain volumes, and the atrophy patterns that you might see towards the bottom there might be represented in something like this, which is easy for, once a radiologist has been trained, to interpret and understand. So the last thing I wanted to mention is about the tutoring stuff that was mentioned very graciously by my colleague Selina. So I just wanted to thank all the tutors out there that work to support students. And I wanted to sort of say, I wanted to encourage people to take on these roles. It can be quite daunting when you move from a departmental role where you're supporting students, where there's like 30 students, you know them all really well, to a faculty level and think, well, I can't go around and know everyone, I can't help everyone, but that's not the point of it. People will find you when they need help. Believe me, they will find you. So you just have to be there. So I would encourage people to do these kinds of roles throughout the university. They're very important and they're very rewarding. So finally, I want to say thank you to my lovely friends from university and from home, and also my lovely colleagues. Thank you.
So it's over to me now to give the vote of thanks to Jo for her amazing lecture. So thank you, Jo, for delivering a wonderful lecture. My name is Professor Tammaryn Lashley. I'm a long-standing colleague of Jo, so it's an absolute pleasure and an honour for Jo to ask me to give the vote of thanks this evening for her professorial lecture. Having your friends and family in the audience and online to celebrate us makes this occasion even more special. And I'm even pleased to see that Dave made an appearance on the slides, because he scares the hell out of me at any virtual meeting I have with Jo, because I'm petrified of cats. So it's nice to see him on the slides. I've known Jo for many years as a colleague and as a friend. We first met while undertaking our graduate tutor roles for then our individual departments, but then later as both graduate tutors in the Department of Neurodegenerative Diseases. This meeting and meetings around tutor roles and our chatting about research sparked various ongoing collaborative studies, some of which Jo's told you about this evening. And our combined expertise of imaging and neuropathology has led to some wonderful collaborations. We've heard from you about your immense amount of work you've undertaken, and you've given us a fascinating tour of the brain using MRI and explaining how the volumetric T1 weighted images provide high quality definitions of the brain. Your work on segmenting and quantifying the hippocampus and your research on white matter hyperintensities has been groundbreaking, and the detailed explanation of how you use these registered images along with the co-registration of the pathology to quantify the brain volume is also enlightening. Jo possesses qualities that are not often seen in academics, frequently giving of herself without expecting anything in return. What not many of you realise is the extensive work that she undertakes as faculty graduate tutor, which has been mentioned by Selina in her introduction, and Jo touched on herself. Her dedication to student well-being and her passion for enhancing the experience at ION and faculty level is truly remarkable. Much of her work at faculty level is often goes unnoticed, and I'm confident that every student and supervisor within the faculty would personally thank her if they knew of all the hard work that she puts on to streamline processes to streamline the processes for both students and supervisors. Working with Jo is a unique experience. She keeps everything real. But she's also the one you can turn to for help and advice. In fact, I've got a post-it note above my desk when I was having a particularly difficult time, something was difficult at work. Jo said it like it is, told me what I needed to do, and now I've got a post-it note above my computer screen that says, Jo said I'll be epic. So whenever I need a bit of support, I know I can always turn to her. It's an absolute pleasure to work with you, Jo, and I eagerly anticipate all the future projects we've got planned, providing we can secure the funding for the projects. I would like to extend my heartfelt thanks to Jo once again for delivering her professorial lecture. She should take immense pride in all of her achievements and in becoming a professor here at UCL, a recognition that's richly deserved. Thank you, Jo.
Great, thank you everyone. So it's now my real pleasure to introduce our second speaker for today, Cath Mummery. Cath is a Professor of Neurology at the Dementia Research Centre at UCL and also a consultant neurologist at the National Hospital for Neurology and Neurosurgery. Cath plays a pivotal role in shaping the landscape of dementia clinical trials, both here at UCL and nationally and internationally. As a leader in this field, Cath has been chief investigator on over 25 early phase drug trials for Alzheimer's disease and frontotemporal dementia. Notably, she's led pioneering work in genetic therapies, with UCL being one of the most experienced centres globally in genetic therapy trials for dementia. Her clinical leadership has resulted in several firsts, including the global first dosing in early trials of gene silencing therapies for dementia. Beyond UCL and the hospital here, she is chair of the NIHR Dementia Translational Research Collaboration and director of the NIHR UK Dementia Trials Network, which leads national efforts to establish a unified trials network focused on early phase clinical trials and the acceleration of dementia research in novel treatments. What I've told you so far, you can see that Cath's ambition is clear. She wants to ensure that we not only have treatments capable of altering the course of diseases like Alzheimer's, but also to ensure that those treatments are delivered promptly, safely, and equitably to those who need them the most. On a personal level, it's a goal of mine to see a fundamental finding from my lab make it through to one of Cath's trials, and we're working on that. In addition to her clinical and research contributions, anyone in the room who hasn't met Cath before might recognise her anyway as the woman off the telly. She's increasingly becoming a well-known media figure, occupying spaces such as the sofa on This Morning, and also in an award-winning BBC documentary, The Jennings versus Alzheimer's. And for anybody who has seen her either there or listens to her tonight, you will see her expertise and her passion for dementia research. And I think it's a service to all how she shares them with the wider public. So please join me in welcoming Professor Cath Mummery to deliver her inaugural lecture.
That was too lovely. Thank you, Selina. Oh, my goodness, how many people are here? They told me they were going to keep it cool. I've got leather on, but it's white clean, so we're fine. Thank you so much for coming and for listening to Jo and to me on what's such an important day for us. so lucky to see so many friends, so many people I love, so many people I admire and I've worked with. So I really appreciate it. This is incredibly special to me. I haven't taken the most direct route to this point, but I think that's fine. I think sometimes there's more than one way to reach success, and there's more than one way to climb to a summit. And the long way around sometimes perhaps is the best way around. At least it has been for me. This really is a story that I'm telling on behalf of an awful lot of people, because the trial isn't done by one person, and my research is in clinical trials. The trial is done by an enormous number of people. Everybody on this picture and more are involved in every single clinical trial that we do in dementia. We have the fantastic Dementia Research Centre trials team, and many of them are here, and thank you so much for coming. I really appreciate it. We also have the clinical team within the DRC that are also involved in making sure patients understand what research is. And then we have the Leonard Wolfson Clinical Research Facility and all of their team. And that's not thinking about the patients and the families as well, and all of the support departments, pharmacy, nuclear medicine, radiology. There are so many people involved in just one trial working. So this story is on their behalf. This is not really my work, it's everybody's work. You may recognize this picture. So this is Nanga Parbat. This is one of the highest peaks, one of the hardest peaks to climb in the world, and it's nicknamed the Killer Mountain. This is somebody I want to introduce you to who has inspired me throughout my life and has kept me going sometimes when things have been challenging. This is not a neurologist. It does look like one, but it's not a neurologist. This is Albert Frederick Mummery. So he is my great-great-great-uncle. And in the 19th century, he became known for scaling peaks and climbs that nobody else had been able to scale before. And he became known as the father of modern mountaineering. And this was his ethos, basically. If you can't do it anyway. And he was known as the father of modern mountaineering as a result of all of these climbs that he did. But he decided to go further than Europe. And he set his sights on Nanga Parbat. So in 1895, he took a team and he started the ascent up Nanga Parbat. He never came down. He's still up there, somewhere on Nanga Parbat. So he didn't succeed in that place, but what he did was he showed the route for everybody else. He showed the path for others to forge. And to me, that's what's really inspirational about what he did. Back to me. So from an early age, my siblings, who are sat down the front here, would tell you that I was quite bossy. Certainly like to tell people what to do. I would say I was very good at delegating, and that's actually really important in clinical trials. So I think I was showing my skills early. I was very passionate about a number of things when I was growing up. One was animals, all forms of animals. I used to have 4 books on animals at any one time at the same page open. I was a bit of a weird child. Because I was interested in everything, wanted to absorb it. And this was my horse, or our horse, my dad and my horse, Bella. I was really interested in drawing and anything creative. I would just do more and more and more of it. And I loved music. So piano and cello and part of a youth orchestra were things that I really enjoyed as a kid. I was going to be, as far as I was concerned, a classical pianist, or I was going to be a zoo vet. It was one of the two. Nothing else was an option. But I don't know if you remember, if some of you were growing up in the 70s, do you remember the halcyon days when people would give children a chemistry set with a Bunsen burner, flammable chemicals, and all sorts of other bits and pieces, and leave them in their bedroom to play with it? Health and safety was brilliant in those days. I wish we were back there, but this was one of my favourite ever Christmas presents, thanks, Mum. And I spent a lot of time playing with chemicals, and perhaps the science side of things was starting to show through a little bit at that point. But there was another factor in what I was going to decide to do. I am a Nepo baby. So that is my grandfather and my grandmother, both of whom were doctors or medically qualified. And my grandfather was a microbiologist. My father became a microbiologist. He followed in his father's footsteps. My mum broke her family's tradition. They were farmers and proud of it. She became a doctor as well. So I had 2 parents and 2 grandparents that were all medical. So there wasn't really a lot of hope for me. But I used to love, on a Saturday, going to my dad's lab, and he'd plate up agar plates, and I'd be allowed to look at them and maybe count them. And they also had a particular smell, I remember. But I absolutely loved it. What I didn't love was the piles of pieces of paper he had to sign off on a very regular basis, and the fact that he seemed to work 24 hours a day, 7 days a week, and lived in a porter cabin. That, to me, was not the most glamorous job that I ever wanted. He was in a porter cabin for a very long time. But he's not with us today, I wish he was, but he was a wonderful dad, and he was also, and you're going to make me blub now, and he was also a fantastic doctor. He was known as Uncle Vic to everybody as a microbiologist. And I did follow him in the end. I didn't go down the classical pianist route. I didn't go down the zoo vet route because there weren't enough of them to guarantee me a place. So I went to UCH, which is where Dad went. And there are some people that I went to university here that I haven't seen pretty much since we went to university. So thank you so much for coming. You can tell the stories later, but I get to tell the stories right now. So this was my year. And yes, Selina's got it first. So there's always one. There's always one. And this was John. Every time. I'm somewhere in the middle in a brown suede jacket. I loved being at UCH. I absolutely loved it. I also loved being at the wider UCL, and I spent a lot of time in social committee in UCL because I really liked to party. So I sort of split my time between medical school and UCL. This is a memory from early on when, do you remember when sponsored hitchhikes were legal or safe? Something like that. So we went on a sponsored hitchhike to Paris, and John is the one next to me on your right. And the 2 of us were supposed to be hitchhiking, and we accidentally cheated because there was a Jaguar that came out of the ferry with 3 people from Guy's who were also supposed to be on a sponsored hitchhike, but decided not to. They just took their own car. So we got in the car with them, and that was our hitchhike. So we had quite a nice time. This is such an 80s photo, and we thought we looked so cool. Look at Adam's fingerless gloves that he's very proudly demonstrating. My goodness. And the hair. Anyway, so, yeah. We had a lovely time. And I'm using the wrong thing, that's why I can't do it. So this is not fancy dress. I know, but it's not fancy dress. So I never really got the memo, less is more. More makeup, more hair, more jewellery, more everything. And that was me going down to the Union on a Thursday for a quick drink, basically. Now, when I was at university, I did supplement my income a little bit, and I did a little bit of modelling on the side. And sometimes that got me into a few scrapes. So this one, I was modelling exercise gear in Olympia in December, because that's when they fashion summer stuff. And the photographer decided that he needed a photo of me on a bike. So some poor unsuspecting passerby on a bike had his bike swiped from him, plonked me on the bike and took this picture. God only knows what position I think I'm in. But the most embarrassing thing about this is where that picture ended up. It ended up in the Reader's Digest magazine with a large headline on the top saying, "What not to wear." What you do for a bit of pocket money, eh? Anyway, so I qualified, surprisingly, and did my house year, and then went to an SHO rotation in medicine in Brighton. And one of the parts of that rotation was 6 months in Housewood Park, which is a standalone neuroscience centre. So this is surgeons and neurologists working together with no other specialties there. And it really made me understand the joy of being in a small-knit group and the joy of being in a specialist group. And I loved it there. It was live-in, so I was actually also doing neurosurgery assistance, which was an eye-opener. But I loved it so much that it really sparked my interest in neurology. And I said as much to them. I said, you know, I think I'd like to do neurology. This was me very new, very young. And the person who shall remain nameless, back in the day, said, don't think neurology is really a good specialty for women. I suggest you do something like dermatology. Didn't take any notice, thank goodness. But yeah, it was a very special place in some respects. I remember some of the gentle wisdom that came from Simon Nurick about you had to listen to the patient before they even got to the door on the way into the clinic, and it would tell you more about their diagnosis hearing their walking than seeing them when they came in. Other people have told me that since. It was well worthwhile. But I decided I didn't want to stay being an SHO anymore. I was in a bit of a hurry at that point in time. And the other thing that I really enjoyed in life was intensive care, anything invasive, really, which maybe given I'm in early phase trials, I know, Selina, anything, given I'm in early phase trials, maybe that's not so mad, because they are quite intensive in terms of what we're doing. So I applied for and took an ITU transplant medicine rotation, about as far away from neurology as you can get. And the first part of that, as a brand new registrar, was being the transplant registrar to Magdi Yacoub in Harefield Hospital. Now, those of you who know who Magdi Yacoub is will be thinking, oh, God. I mean, he was an extraordinary man, but he was also driven beyond belief, very demanding. He used to do clinic appointments that would be at 3 a.m. on the letter. The patient would check, and they'd say, yes, that's right, it's 3 a.m. that you need to come for your clinic appointment, because that's the only time that he has free from operating. He was a very inspirational man to work with. And what they really showed me in Harefield was the combination of research, cutting-edge research, and clinical happening completely as one. I hadn't seen that before, and they had totally embedded the two. I also had my first experience ever of research myself. So that picture next to Magdi Yacoub is Abe Guz. I don't know if any of you ever met Abe Guz, but he was a larger-than-life character professor of medicine and was an absolute scream. He was one of the funniest men. He was brilliant. And I remember we used to wheel patients after their transplants, their lung and heart transplants, through the car park in a bed to the respiratory lab. And he would end up adding a bit of a Wurlitzer in the middle of this because the patients enjoyed it and he enjoyed it. He was bonkers, but brilliant to work with. So I survived Harefield and went to Central Middlesex for the next year. And I was lucky again because in those days, in the intensive care unit, where I was now a registrar, the lead consultant was Derek Bell. Now, Derek Bell is now very, very high up, and he's the Royal College of Physicians Scottish president, and up in Edinburgh. He's one of the best leaders I've ever worked for. He brings everybody with him. He taught me a huge amount about how you get people to follow what you do, how you bring people with you with ideas, you don't push them. And he was fantastic to work with. And the other person I worked with was Richard Wise. So back, really, to neurology, even though I'm in the middle of ITU and respiratory medicine. And Richard is no longer with us, but he was a fantastic cognitive neurologist, and he was very interested in language and semantic processing particularly. And we started doing ward rounds together, and we started doing clinics together and talking about his interests and my old interest in psychology from when I was a student. And eventually we did some pilot work with functional imaging with PET, looking at semantic processing, meaning processing in the brain. And that led to an application for an MRC clinical fellowship. And that really was a bit of a reawakening. But it was also, and that's me on my first parachute jump, it was also a bit like jumping out of a plane without a parachute, because I was going from being an intensivist and a registrar rotation to a very hothouse environment, which I'll show you in a second, that was cognitive neurology, PhD, and away from patients completely. And I think at the time I did question a bit as to whether I was entirely mad or stupid or both to do this, but it was one of the best decisions I ever made. And so I went from that rotation, I left it early, and went just across the road to the functional imaging laboratory under Richard Frakowiak at that point in time. And I arrived full of enthusiasm and full of trepidation, because I really did not know very much at all, and having been recruited by Richard Wise. Now, this was a completely different environment to anything I've ever seen before. This was a multinational grouping of people across physics, radiology, neurology, psychology, philosophy, all of the people you'd want to talk about what on earth is happening in a brain in one place. And it really blew my mind, and it showed me something around research that I'd never seen before. I was really lucky, again, that I had John Hodges and Carolyn Patterson from Cambridge. as collaborators, and they were beyond patient. They taught me everything that I know, or at least knew then, about neuropsychology and clinical patients. And they had a big cohort of patients with semantic dementia, so we also had some down here, and they were amazing to work with. But the luckiest break for me, who's sitting just over there somewhere, she's going to blush hopefully, was that I got to work with Cathy Price. And Cathy eventually became my primary supervisor for my PhD when Richard left the Phil. Cathy probably is the reason I got a PhD. She is an amazing woman who is driven, but is also an amazing mentor who will sit you down and pull you through the bits where you think you can't do your PhD anymore and show you that you can do those things. She's an unbelievable presenter and a very good teacher, and I absolutely loved working with her. So thank you, Cathy, for getting me through that bit. Yes, she's blushing. Good. It was an amazing time. We were a really close-knit group, and we had a lot of fun. We also worked very hard, and you'll recognise some of the people in these pictures. These are all from PhD. So there's Carl on the left in the top, then there's Eleanor Maguire, then there's Cathy. You can see our Vice Provost is in a number of those pictures, and it's interesting, he's got a skill that nobody knew he had before. So he's very good at eyelash curling, as he did on Sarah Jane Blakemore in that bottom middle picture. And the person you can't recognise on there, by the way, was the best-dressed skeleton at the Fancy Dress Party, and that's James Rowe, for those of you that know James Rowe. He does like a party dress-up. But we did work at the same time. We weren't just playing. This was my first ever paper, which is why it's there, because I think the first ever paper you write as a first author just makes you go, oh, I did it. And we could only afford black and white pictures at that point in time, so that's why that looks like that. But really, my PhD was about trying to disentangle what happens in semantic processing, both in normal people and also how it's disrupted in diseases like semantic dementia. And we did a number of papers where, again, I was really fortunate to work with people like John Ashburner just when we started doing voxel-based morphometry, just trying to really pin down what was happening precisely in voxel language in the brain, and then correlating it with the cognitive change. And I worked with some forces of nature. Sophie Scott, I don't know how many of you know her, who's the director of the ICN, is a fabulous woman in every way, and was at that point interested in language. Now she's much more interested in humour and the science of humour. But she was fabulous to work with. Tim Shallis tried, and I think probably failed, to inject some sophistication into my modelling of semantic processing and the designs that we did, but was fantastic to work with. And Eleanor Maguire was just all-round brilliant. So Eleanor is sadly no longer with us. We lost her very recently to cancer, which is a bloody disaster as far as neuroscience is concerned. She was an incredible neuroscientist, but she was also somebody with a very wicked sense of humour. And we did this one study which was trying to look at the interaction between semantic memory, so meaning of things, and episodic memory, so events, and autobiographical memory, my own memories, versus public memory. So we had a whole load of conditions in the PET scanner that we had to write sentences for. And, you know, it might be something like, you have 4 children with brown hair, or it might be, my daughter's name is Astrid. And Eleanor left one of the sentences in that perhaps in hindsight we should have taken out, which was one about public knowledge, which was, is the Pope a Catholic? We kept it in. There was a lot of movement artifact in that particular condition. But it was amazing working with her. I learnt a huge amount from her. So I got my PhD and then went back to, and I told you it was a long road round, then went back to full-time neurology registrar. So now I'm a registrar again, but this time I'm a neurology registrar. And I was at King's and Queen Square for the time that I was doing this. And a number of people really kept me on the straight and narrow and taught me the little that I know now. At King's, Nigel Lee and Lena Nashef were unbelievably patient with the very green around the gills person that had turned up, which was me and didn't know anything. Bridget MacDonald, who was my peer there and is here now, and Katia, were incredibly patient with me and used to take me for a lot of cups of tea after ward rounds. Tea. And the person in the profile is not there because I could not, for love nor money, find a picture of him. It's George Harwood. George was the best cognitive neurologist I had ever met at that point in time. What he didn't know wasn't worth knowing, and none of it was in books. And he was the most gentle, wonderful man to work with, and I miss him. So I learned a huge amount at King's. And at Queen Square, again, I learned an enormous amount, and some of the people are either listening online, Andrew, or here. I think Peter is up there. Mike, Hannah and I used to do ward rounds at the Middlesex, when Middlesex used to have patients in it. And he was always quite amused by the fact that I had a surgically attached can of full-fat Coke at all times, even when seeing patients. Peter Rudge is the most incisive diagnostician I have ever met in my life, and also has more energy than most 25-year-olds, and you have to like a joke that's slightly non-PC. Andrew Lees was the person that taught me you did not have to think the same thing as other people. You did not have to go with the flow. You did not have to accept dogma. And if you didn't listen to your patients, you were a total waste of time. That was the most important thing. Listen to your patients and do things the way you want to do them. And Mary Reilly, MBE, and I'm trying to make you blush now, I was a little bit scared of Mary when I first came. Mary has been the biggest support to me throughout my career. I think bar none. She has always nudged me in the right directions very gently. She has always told me that I can do the things that I've doubted myself about, and has been an unbelievable mentor. Thank you, Mary. So that was my training. And then I got finally to be a consultant in neurology. And I started at King's and I was happy there for a couple of years. But then I got given the opportunity to come to Queen Square. And the reason that was so attractive was because the Dementia Research Centre was part of the deal. And I would be able to work there with Martin, who set it up, and his right-hand man, Nick, who now leads it so beautifully and collegiately. And how could you say no? This was the best centre in town. This was the place to go to try and find and understand what was happening in cognitive neurology. And so I was really lucky to be able to take up that role. It was a full-time clinical role, though, so there was no research at this point in time. So I tended to do things that were more clinically based, leading the dementia service, trying to develop that, and also with Mike Hannah's support and Mary's support, taking on roles like the Associate Clinical Director of Neurology and trying to improve the clinical management in the hospital. One of the things that I did as part of the dementia service changes, and I'm really delighted to have the team here, they're just over on the right, was to develop a cognitive clinic for those that are profoundly deaf, that are BSL signers. And this is because these people cannot access decent, and even if they do access decent services, they are not dealt with in the right way. We don't have the tools to be able to assess them because they're not validated for people that are BSL signers. And Joe Atkinson and others have developed tools that are able to use those. We use them in clinic. We did a clinic this afternoon so that we actually give people a better experience. The number of people that have got diagnoses that are either late or different what they've been told before is humbling. So I'm really proud of that service and proud of the team that are such strong advocates for support for those people. And the one thing that actually, Mary, yeah, you were involved in this one as well. So, Chair of the Services Committee, I got very involved in the ABN. And this, again, was partly because I was very involved in clinical and in management, and this was taking that onto a national role. Now, that was a lot of hard work. Anybody that's worked in the executive, the senior level of the ABN, the Association of British Neurologists, will know there is a lot of work in what you do. However, the reason you do it is because it's so much fun. The people that work in the executive of the ABN are a fabulous group of people. And basically, it doesn't matter how hard you work if you're enjoying yourself, which is actually one of the things Martin Rosser told me many, many moons ago. If you're enjoying it, keep doing it. If you're not, stop. So we did a lot of work trying to really optimise the conferences for the ABN, the support for neurologists. And that came to a head really within the pandemic, which was part of the time while I was doing this role. And there was a huge amount, you remember how much was going on. But if you were a neurologist, you were often being farmed off to go and do other things. You weren't in your role anymore. Things changed enormously if you were a clinical neurologist. And what we tried to do was support people in those changes and give them guidance about what they could and couldn't do or should and shouldn't do and how they could deal with patients remotely with a neurological problem. And we didn't do a perfect job, but we did try very hard. One of the people that worked so well with us on that was Bod Goddard, who's that picture down the bottom, who was the president of the Royal College of Physicians at that point in time and did an amazing job of trying to corral all of the different specialties together, as well as NHS England and the Royal College. So it was a fabulous 6 years being a part of that executive, but I did start to think maybe I should be focusing a little bit on doing some more of the research that I'd said I always wanted to do. But before that, a brief interlude. So, 2012, I got an email in my inbox, and it must have been a quiet day because I looked at it rather than binned it because it was automated. And basically, it was asking for volunteers for the Olympics. So many queues later, auditions, waiting around, all sorts of other things, I ended up being a dancing nurse in the Olympics. And I'm pretty much, I'm just right and slightly below centre in this picture, so you may even be able to see me. But this was an extraordinary thing to do. There were 150 plus hours of rehearsals that went into what we did over a 6-month period for a very short segment. But it was the most fun. It also taught me a huge amount. I am not a patient person. Stop laughing. I'm not a patient person. And the thing you had to be to do this was very patient, because something that's got 4,000 people in it, which was the total volunteer level for the whole of the ceremony, you're spending about 5% of your day, which is a 10-hour day there, doing something. The 95% of the time, you're waiting to do something. So boredom was a part of it, but also teamwork and working with people in something that was the most unbelievably positive thing. I, by the way, am in the third arm of the N, swinging around a bed with a very lovely small child on it. That is me on the way to the actual opening ceremony, looking quite matronly, I have to say. And that's us after the opening ceremony, on the way to the pub. The pub was very interesting. There were about 150 nurses in it. And one guy actually came up to me at the bar and said, "Is there something going on?" So, research, finally. So I started doing trials because Martin Rosser came to me one day and said, "I just wondered, would you be interested in doing this?" And he talked to me about a particular trial that they'd come for interest to him. He didn't have time, clearly he was doing a lot of other things. And I thought, well, it involves patients, it involves trying to find treatments for those patients. It's got a lot of checklists and stuff like that, and I'm slightly obsessive, so I think I'll quite like those. Yeah, why not? So we started trying, and Jane is in the audience somewhere, who was the senior research nurse at that point in time and was the only research nurse. So there was me with an hour a week and Jane, and no place to do these trials. It was a nightmare. I think it took 2.5 years for us to fail to get the first trial set up. Very hard work. Part of the reason was you need a lot of time and you need a lot of people to be able to do clinical trials. One person in an hour a week isn't going to cut it. And it took me a while to get the time. The blue levels, by the way, are research time. The orange is clinical. So by 2018, I was around 50-50 in terms of clinical research. But prior to that, for a very long time, I had very little time in research. The game changer was this. And thanks to Nick and thanks to my Hannah and others, there was a significant donation from the Wolfson Foundation, and part of that went to build the Len Wolfson Experimental Neurology Centre, which is just next door. And this opened in 2014. And this was a cutting-edge, bespoke, neurodegenerative specific arena for us to be able to do early phase trials in. And Vincenzo Libri was brought in from Imperial to head it up, and we imported rather than poached, I hasten to add, nurses from UCH cancer early phase trials, which Maravik, Kerry, who's not here, and the lovely Edwina, who's now in Australia, please bring her back. And that gave us the gravitas and the credibility with pharma and biotech to say we can do these studies. And that really changed it. So that by now, this is what the team looked like in my trials unit. We now have 19 people in the trials unit across a number of different disciplines. And this is what's critical to do a trial properly. If you don't have this sort of staffing level, it is really hard to make it something that withstands the flex of activity in trials. And many of them are here. I'm not going to name them all because I don't have time, but thank you, you know who you are, and you know that this is down to you as much as anybody else. So I just want to start talking about my research itself by coming back to patients. I'm a clinician first and foremost. Patients are the point of everything for me. And our patients give us the questions, but they also give us the answers if we actually listen to them, like Andrew Lees would say. This patient did more probably than most AD academic researchers in terms of impact on Alzheimer's disease in the course of it. This is Carol Jennings, and you heard Selina mention the Jennings documentary. I'll mention that again later. But Carol in the mid-80s heard that a very youthful John Hardy and a very youthful Martin Rosser were looking for families that had a lot of dementia in them. Their people were dying young of dementia. They were trying to find genetic causes for Alzheimer's disease. So she wrote to them. She said, you need my family, and I will bring them. So she did. And she got all of her family to have blood tests. And as a result, the first genetic mutation to cause Alzheimer's disease was identified by, I'm sure he looks like Damon Albarn there, by a very young John Hardy. He looks very cool. Anyway, that really changed the game because we then had the amyloid hypothesis that John put together with others, where basically Alzheimer's disease, we're thinking it's caused by early triggering build-up of amyloid protein. And so trials were going to start focusing on trying to remove that amyloid and stop that happening. And that happened for many years, but just for those people that are not neurologists, very, very briefly, amyloid plaques are the first thing we think that builds up in the brain, but it's not the only thing. There are also abnormal proteins called tau that build up in the brain alongside the amyloid and seem to be spread and accelerated by a particular level of amyloid. And then the immune system has a modulating response to the toxicity of those two proteins. So there are many things that happen in Alzheimer's disease, not just amyloid, but that is what we think is a very early part of this. And for many, many years, there was a negative after negative after negative news flash on the different trials that occurred against amyloid. People started to think maybe the amyloid hypothesis was wrong. But we now have 2 fully approved drugs that remove amyloid from the brain. They are being used in clinical practice in USA, in Japan, in a number of other countries. Lecanemab and donanemab both decrease amyloid levels in the brain, remove the amyloid from the brain enough to slow down cognitive progression. But they only do it by about 30%. And there are potential significant side effects. So these drugs are not perfect by any stretch of the imagination, but they are a beginning for us to build on. So this was a huge moment for all of us in terms of you can change the course of Alzheimer's disease. We didn't think you could, or we didn't know whether you could, and now we know you can. But you can only change it a little bit at the moment, so what can we do to make it better? Where do we go from here? Well, we knew, and John Hardy and I looked at this, and others have as well, we knew that if you look at the different trials that have been done with anti-amyloid drugs, and these antibodies that are given in infusions, it matters how low you get the amyloid, it matters how long the amyloid is low for. And so it seems that if you drop the amyloid, and centiloid is a standard measure of amyloid, so if you drop the level below around 20, the studies are positive. If you don't get it that far, over that period of time, the studies are negative. So can we accelerate that amyloid removal? Can we reduce those potential side effects that I was talking about at the same time? For a very long time, we've been relying on drugs passively crossing the blood-brain barrier, the barrier between the brain and the rest of the body. And that means 0.1% of the drugs we give actually get to where you want them to get. That seems daft, doesn't it? What do you want to do? Well, you want to really take the drug across the blood-brain barrier actively so you get more drug in. That is exactly what is now being done. The methodology is being developed to transport these molecules across the blood-brain barrier so that the drug gets to where it needs to get much more efficiently and much more quickly. And we've been trialling a drug called trontinemab, which is basically one of those anti-amyloid antibodies attached to an active transporter across the blood-brain barrier. That trial is ongoing, but these are the interim results. And so if you look at this chart, what you see here with the top dose is a rapid reduction in amyloid by 3 months. They've had 2 doses of drug. That's the same result you saw after 18 months in those drugs that are now approved. So you see a significant acceleration of reduction, and it gets very low. So these people are effectively negative on their scans, if you looked at their scans. But does that cause problems with side effects if you're doing it so quickly? Well, the answer is no. Because of the way that these drugs get into the system being slightly different, they don't seem to cause the same levels of side effects as those previous drugs. So we are improving things potentially already on the basis of those first drugs, which I think is really exciting. And I think this will really help with other drugs, getting them across into the nervous system as well as just the antibodies. But are we giving them at the right time? Well, amyloid deposition, I think we heard earlier, amyloid deposition starts in the brain way before you have symptoms. By the time you have symptoms, it's been there around 20 years and you've got a head full of the stuff. And we know this from looking at people that have a genetic cause for Alzheimer's disease. We can see from when they're predicted to get symptoms, we can see the amyloid build up earlier than that. And so for a long time, and some of the people in this room have been involved in this study very, very closely for a very long time, for a long time now, we have been studying that in people that are at risk of genetic Alzheimer's disease. And so the first drugs we tried in this platform of people that are at risk of genetic Alzheimer's disease, they haven't got symptoms when they go into the study, were 2 of those anti-amyloid therapies. And they were on that for 4 years in total each, but actually it was a common close. So basically some people were on it for 7 years. That's a long time to be on drug. It didn't show an effect on those people when the initial results came out clinically, but it showed a biological effect. It looked like it changed the markers of disease, and Joe was talking about some of these earlier. And it was felt that if it did change those, perhaps there were other reasons that we weren't seeing a clinical result in these patients, and therefore why didn't we continue to treat them with drug, all of them, and see what happened, which is exactly what we did. So they've now been on treatment for at least 12 years. Now, these are very small numbers of people, so take with a pinch of salt. But what happened when these patients in the asymptomatic group, no symptoms, were given treatment for this long, you looked over that 10-year period, their risk of developing symptoms halved. So it looked like the drug was helping delay onset significantly. And this is something, obviously, we'd want to look at further. But that is a very promising result. If you give the drug early enough, perhaps you do get these sorts of effects. In addition to that, we thought, well, if we're going to look at secondary prevention, when people have disease but are trying to stop the symptoms, why don't we go even earlier? We know that people with genetic mutations are going to get disease at some point if they have a mutation. So what about before they have disease? We try these drugs to stop the accumulation of amyloid at all. And that's exactly what we're about to start doing in the coming weeks and months. There are some important pointers about this. These people are 25 years before symptoms. They're in their early 20s. They might be in their late teens. These are really young people they may have young families or be thinking about starting a family. They may be just getting their career up and running. So it's critical that the way we do this is done for patients, the way that it suits them. And there's been a lot of discussion about how best to do that, and I think that's absolutely critical. Don't forget, this is genetic disease, but these sorts of results very early on could potentially inform us as to what we could or couldn't do in people with sporadic disease. It's too early for that just yet, but I think it will help us understand the picture better. I'm going to leave anti-amyloid antibodies now because we have got other things that we have been focusing on. And gene therapy is one of the things that Selina mentioned that I have been focusing on for a number of years. And we can try and target amyloid in a different way. So I've been talking about anti-amyloid antibodies that mop up the protein once it's already been deposited in the brain. Well, why don't we work upstream of that? Why don't we try and stop production in the first place or decrease production in the first place? If we think that young onset Alzheimer's is potentially an overproduction problem, would that be a sensible thing to do? And so interference RNA is a gene silencing therapy that tries to interfere with the process of the APP gene being transcribed into a message, and that message being translated into the amyloid protein or protein components of APP. And so this interference RNA does that by hijacking the cell. And we started doing this a couple of years back. And this, again, is a study that's ongoing. And thanks again to the team who have been, and the participants, because there are some actually that are either listening in or here, that have been part of this trial. And so we have looked at one dose of this drug, and these genetic therapies are given intrathecally because you can't give them as an infusion yet because we're not using the active transport mechanism with them yet, but watch this space. And there are a number of doses that we have given as a one dose to people with young onset Alzheimer's disease to see, firstly, is it safe? Secondly, does it drop these levels of protein? And the answer is yes. So what we've seen so far is a really dramatic drop, really quick drop in the levels of protein fragments of APP in the spinal fluid. And this goes out, this is small, so you probably can't see it, but it's sustained out to 12 months. One dose has an effect that lasts 12 months. So you wouldn't necessarily have to have an intrathecal injection every week. This would be maybe once a year, which is a very different prospect. Related to that, we saw changes in the precursors of amyloid, which are the bits that build up those plaques that I was talking about before. So again, very promising, but very early results. Importantly, they don't cause the same side effects as the anti-amyloid antibodies. So those people where they're dangerous, and there are certain groups that have a higher risk, this could be an option. So lots more to do, bigger study to do, and we're currently in the multiple dose part of this to see how that plays out. But we need to go beyond amyloid. I said it's not just amyloid, there's tau as well, and Selina's favourite thing. So we know that tau plays a critical role, and we know that you don't have a head full of tau when you develop symptoms. The symptoms correlate in severity and spread with the level and spread of tau in the brain. So it makes sense for it to be a good target to try and attack. We also know that tau and amyloid have a reciprocal relationship, so they impact each other in their toxicity, and that the tau spreads by seeding from cell to cell. So if we could try and stop that seeding and spreading and try and reduce that tau level, potentially we could reduce tau and amyloid toxicity. Two methods I want to mention to you briefly. The first, I think this is unique, and this is thanks to a friend and colleague of mine, which I'll come to in a second, but there is a collaboration between Eisai and there's some people here, I think Charlie's here, from Eisai and UCL, in terms of a number of projects. And this is one of the projects, making a drug which is an anti-tau immunotherapy. So like the anti-amyloid immunotherapy, mops up tau, not amyloid. The aim is that it gets to the tau that's spreading between the cells and prevents it progressing. There have been a lot of negative trials in this area so far, but perhaps that's because they've been targeting the wrong bits of tau. And Rohan de Silva invented, in collaboration with Eisai, this drug, E2814, which targets a different part of tau. It targets part of it that is required for it to do that seeding between the cells. And we started a trial that we led from UCH in this in September 2021, just after the pandemic. Great time to start a phase 1 trial in people with mild to moderate Alzheimer's disease. And we completed it last year. Now, this is a tiny trial. We got 4 patients in, and there were another 3 patients across 2 sites in the US. That's it. 7 patients globally in the trial. It's minute. But it did show some interesting things. It showed that perhaps this did do what we wanted it to do, that this tau immunotherapy was getting to enough tau to actually drop levels of the pathological tau in the CSF. So it's now in a bigger trial, back to the DIAN-TU platform in people at risk of genetic Alzheimer's disease. We're using that drug in a larger number of people, 168 people, in combination with one of these anti-amyloid therapies, because the world has now got much more complicated. In the States and in other countries, anti-amyloid immunotherapies are standard of care. So trials often have to have that treatment in them as well as the new drug you're trying to look at. Life gets more complex, but potentially it gets more efficacious as well. The second tau drug I want to mention is again a genetic therapy, and this was the first drug that, and you mentioned this one, Selina, this is the first drug that we trialled a gene silencing therapy in dementia. This is similar to the interference RNA, but not quite the same. It's basically a synthetic oligonucleotide, a single strand, that binds to that message, the messenger RNA that comes from the gene, in this case the MAPT tau gene, and then directly results in its degradation, so it doesn't get turned into tau. So you reduce the levels of tau. In the mice, and I know they're mice, not humans, but in the mice, what we saw in red was decreased levels of tau deposition, improved survival rate, and this is a standard survival rate as opposed to the other where they are surviving or not surviving in red, and normalization of nesting behavior. And on this here, it showed a reversal of tau deposition that had been there before when they were younger because they had the drug. Potentially it reversed the disease, potentially. So we put it into humans, first patient in 2017. That was a long time ago. The only person that had done anything like this was Sarah Tabrizi, had done a trial just before this in Huntington's disease. Both of us were really quite terrified about what we were doing, giving gene silencing therapies to people with Alzheimer's disease. Now look where we are, we're doing it all the time. So this study was in mild Alzheimer's disease, giving multiple doses of either drug or placebo intrathecally and looking at increasing doses of the drug. And the reason that Roger Lane is up there, that's the guy at the top, is because he was really uncertain about whether we had the capability to do these studies at that point in time. This is a long time ago. And because he'd done some stuff with Sarah Tabrizi, and because he came to the unit and he met me and he saw what the Leonard Wolfson could do, he took a punt. Now, thank God for that, because on the basis of that study, we now do more studies in genetic therapies than anybody else. But it was thanks to him for trusting us to be able to do what we said we could do. And he was also an incredible colleague all the way through this study, from start to completion. We gave people long-term extensions, so they all got drug after a certain particular point in time. And the results are basically, if you look at the left-hand graph only, because they're virtually identical, this is placebo versus active. Placebo is the horizontal line, and this is a dose-dependent reduction in levels in the spinal fluid of tau. So it's reducing levels of tau in the spinal fluid. When you give everybody drug, everybody goes down to about 40% of their baseline level of tau. So it's doing what we wanted it to do. The question is, does that do anything to the brain, like it did in the mice? Very small numbers, so again, it needs to be replicated. But what we see is that it looks like it does. So across all of the brain regions and across all of the different groups, once they go onto drug, they show a reduction in levels of tau in the brain on a PET scan that's most in the areas where you would expect there to be most tau. And if you look at two examples, what that looks like is these two people at the beginning on placebo, so they've got slightly more tau. Tau is bright red and green in the temporal lobes. They go onto drug and it clears up to an extent. So this has never been seen with any other drug. What we need to do is show that in a bigger study, which is really exciting and is currently ongoing. Last drug I want to talk about is the immune system. So this is something that's revolutionized cancer. You all heard of checkpoint inhibition being used in cancer therapies and what it's done to really enhance clearance of cancer cells. Well, why don't we try doing that in Alzheimer's disease? And so Michael Schwartz and others have done some amazing work looking at what happens if you rejuvenate the immune system using a checkpoint inhibitor peripherally. Can that change what happens in the brain? And their preclinical work suggests that it increases traffic of specialized cells to the brain, which increases clearance of these proteins and therefore slows down the disease. That's the theory. And the current study is ongoing. And again, these are new things to be doing in this patient population, so we were really a bit trepidatious about it. And what we did was we asked a whole load of oncologists who are very used to using these drugs all the time to come and sit on our safety monitoring board. And actually, this has been remarkably safe. So we've recruited fully to this trial now, and we've had no serious adverse events that are drug-related, which is amazing. We've had a few minor immune-related events, but it's gone incredibly well and continues to do so. So I wait to see what the results of this are, and I really hope that we're going to get something positive from this. But again, it's a very novel methodology, so you're not going to win every time. So I think we've increased the effect of anti-amyloid therapies, potentially looking at brain shuttle. We're looking at combination therapies and prevention therapies. And we're looking at a range of targets that's much wider than it was before. But if we do all that, and similar to what Joe said earlier, and we can't give the drugs to patients, what's the point? I mean, that's a tragedy. If we've got drugs and can't give them. At the minute, in this country, we can't. That needs to change. So we need to redesign our services so we're able to do that. And we need to be working collaboratively and internationally so that we all understand what we're doing with these drugs. If we do that, we'll be able to tailor medicines to the individual in the future. And that's where I hope we're going to get to. Brief thing about redesigning services, and this is work done with Nick and Jeff Parker and Miguel Grillo is our PhD student on it. This is how many scanners we've got compared to everybody else in the world. It's rubbish. We're bottom of the league. Eleanor would say Crystal Palace were usually down there, I think. So, ultra-fast MRI. If you've got a standard MRI process or clinical process, which is there on the left, cognitive and clinical assessment, a 30-minute scan, and then either a PET scan or a CSF examination gives you a diagnosis. That takes a long time and a lot of money. So can you do a quick scan, much cheaper, much easier to access, you can get more people in the scanner, a blood test, and make a diagnosis as well as you can with all of the other things? And that was the question that we were trying to answer with this study. And the short answer is, in terms of diagnosis, really can't tell the difference. In terms of monitoring for those potential side effects, very difficult to tell the difference. And this has been shown by Miguel and the team to be non-inferior in a particular use of scanner. So now what we're trying to do is roll it out in all sorts of other scanners, because there are all sorts of different kinds of scanners, and talking to drug companies about how we might be able to use this to really enhance access to scanning for diagnosis and for treatment monitoring. And I think that's a really practical thing that we can do. The last thing in my last couple of minutes that I want to mention is around the national way of looking at this. So I've talked a lot about what we've done at UCH and how we work with a whole load of people nationally and internationally. But what are we doing in this country from the point of view of early phase trials? If you look at this map of phase 1B trials in Alzheimer's disease, it looks like the states have got it absolutely wrapped up. It's not true. If you drill down into it, and this is now as opposed to 10 years ago, there would have been none in the UK. Now the UK does outperform everyone in Europe and most states in the US, with the exception of the ones where I think the average age is probably the age that you want to go into a trial. So they may have an advantage there with their population. So we have the expertise, but we don't have the capacity. We need more of the good stuff. And we need to teach from the areas with expertise around the country. And the government listened to this and so have funded for £50 million, a 5-year programme to resource 20 sites across the country to do exactly what I've just been telling you about, to do those sorts of trials in the right way, in a harmonised way, across the whole of the UK. UCH, UCL, UCH will be the coordinating centre of that network, but this is a federated network. This is not mandating things, it's a facilitatory network to try and just ease the path of how we do these studies and adapting to what industry needs, what the ecosystem needs, and what those sites need. There'll be a lot of training so that we really do bring on future leaders in trials and provide career progression, because at the minute being a trialist is really difficult to get funding for. And there'll be a participant support framework, which is absolutely critical for those people that spend all that time and commitment coming and doing your trials for you. And there'll be an Alzheimer's Society Research Nurse Network embedded within this to take this really to the local communities. And this is the leadership team here, Christine Chow, who's in the room, who is refusing to show her face because she's miles up on it looks like you've got your sort of hand of God, Christine, in the picture. So there's 2 of us parachuting. There's the long-suffering Ali, who is my amazing PA, who finds spaces in my diary nobody else does. And there's wonderful Claire Rooney, who's the Alzheimer's lead research nurse and DTM research nurse. And then the very handsome Seb Crutch and JP Taylor. We get the mad pictures, they get the handsome pictures. But these are part of the leadership group that are really trying to pull this together. But it's being pulled together in partnership with industry. It's critical. We listen to them, we work with them, and it's a partnership, not an us and them. And we have many partners, and we will have many more partners, and we liaise with them to try and work out what the hotspots are that we need to try and improve. Because what we want is a community, regional-based network that does things as one, that improves access to every patient, regardless of where they are and who they are, and that develops that future-proof leadership and career progression. And finally, we have to partner with the right people, not just industry. And I'm delighted that Hilary is here, who is one of the co-chairs of the Dementia Goals Programme. And we partner with them to make sure that the wider acceleration of therapies is something that we are really looking at. She's also the CEO of AI UK. And AI UK are one of the biggest funders of work in this area. So it's critical that we talk to the other people out there and internationally. You mentioned the Jennings versus Alzheimer's. If you haven't seen it, it's a hard watch, but it's an amazing watch. The Jennings family are an incredible family, all of them. And they're all still involved in advocacy for research. But the media have got a grip on the need to raise awareness recently. These were all in the last year. And that's fantastic, because if we don't promote awareness, we're not going to get diagnosis, and we're not going to get people into clinics, never mind trials. And you mentioned the sofa on this morning, which I've got a picture of. But I think Martin Frizzle is here somewhere, and I wanted to thank him, because he really has promoted, as the executive producer until recently, he has promoted dementia awareness on that programme, which has a huge audience. So I think this sort of thing we have to do more of. I mentioned Carol and John, who are up the front and centre, but there are so many amazing patients and families that are involved in our trials, and this is just some of them. And it just reminds me, when we talk about dementia, we're talking about real people. They're all people that are smiling, that are doing things, and I think that that's incredibly important. Sometimes dementia just looks like a dry word on a piece of page, and it's the opposite to that. So I'm finishing now. I just want to thank my parents. I said it's sad that Dad isn't here. I'm pretty sure he's up there having a glass of champagne laughing because we haven't got one yet. I want to thank my mum, who is in the front here with my lovely godmother, for being the person that has supported me for everything that I have done and said you can do everything, you've just got to put your mind to it. My lovely, noisy, eccentric family who keep me grounded at all times. My wonderful friends, many of whom are here, thank you, and thank you for staying the distance as well. The gorgeous Roman, who, by the way, is now a total diva because he got a cameo in Jennings versus Alzheimer's, and he has a best side, apparently. And my lovely partner, who is not going to blush, he's sitting there saying, I'm not going to blush, who has kept me sane, and has given me the most amazing advice throughout. He's the calm one in the relationship. He's the one that when I've gone a bit doolally actually just laughs at me, gets me to calm down. So thank you, Bob. Love you. Thank you for looking after me so well. I couldn't do it without you. So it's been a long old road, but it's been an absolute blast, and long may that continue. Thank you very much for listening.
Well, thank you very much, Cath. I'm delighted to be the person to be asked to do the vote of thanks. I'm Mary Reilly, I'm Professor of Clinical Neurology and Head of the Division of Clinical Neurology in Queen's Square Institute of Neurology. So I've known Cath since she was a junior doctor here and seen her to go through many things, and I thought that was a typically beautiful, eloquent, informative and entertaining talk. Just right for an inaugural. There's a couple of things I wanted to say about Cath. So the first thing is, they're not bad things. Queen's Square was founded in 1859. So it was and is a hospital for patients. The Institute was founded in 1950 by the hospital consultants to do research. So this place is all about translational research. And I think both talks tonight exemplify that. But I think that's really important. But life has changed such that it's very difficult to do both clinical and do research if you're clinically trained. And Cath is an example of someone who was 100% NHS. Her jobs were busy NHS jobs, as you saw on her diagram. And then with lots of hard work, she merged into a clinical academic, culminating in getting her chair this year. Not only did she do that, but she did that through clinical trials. Now, she hasn't really said how hard clinical trials are. I don't know who's been involved in them, but they're beyond difficult to get up and running, particularly when you have no team in the beginning. And, you know, drug companies don't fund in advance, they fund as you recruit. So it's almost impossible to get started. So to build your academic career in clinical trials is firstly difficult, but then to take the innovation of doing phase 1 and genetic therapy trials was extraordinary. And I think there's huge congratulations to have done it that way. Now, while she was doing all this, she was also building up her leadership portfolio, as you've seen. And I worked very closely with Cath, having encouraged her to apply to be the chair of the service committee in the ABN when I was on the ABN executive as well. And what she was phenomenal doing that job. She remapped the whole of neurology through the UK to see how it could become more efficient. And we called them Cath maps. They were literally Cath maps that were used to actually redevelop how neurology was delivered. So I'm not at all surprised to see her now in the director role in the NIHR with various things she has talked about. But what people don't realise is because she was so good at that job that people tried to poach her. So she was offered to go to fairly senior roles in the NHS. And this is where she was true to herself. There was multiple decisions she had to make, and we discussed many of them, really attractive, that would have been really good for her in her career. But her goal was to be in dementia research and to find a way of trying to drive that forward. And this is at a time when there were no therapies. So it wasn't like she could see ahead and think, I'll be running clinical trials in it. So I think for sticking to what she really wanted to do, it is very, very admirable. And the final thing I'll say is, you know, her advocacy role. And this has been mentioned. You know, we laugh and say it'll be Cath on the BBC News tonight when there's a new drug. But actually, it is really important that there is somebody on the news that actually is eloquent, that knows the drug, but is also very balanced, because it's very tough on the community with any disease.